Okay, welcome back. Today we are going to talk about human chromosome ideogram. In previous video we talked about karyogram, and then we talk about what is meant by karyogram versus karyotype, the difference. Karyogram is the diagram, karyotype is the one we write as the answer. Then about ideogram. This has higher resolution than the karyogram. We can identify banding pattern and the length of the chromosome clearly compared to the karyogram. Ideogram is better. Here we use GMSR staining method, and you see there are light areas of banding and the dark area of the banding. What do you think? Which areas contain higher genes? Okay, light area contain higher gene. Rich in genes are the light area. Poor in genes are dark area. Okay, and usually the length will be meaning the number of the base pair, because more and more, when there are more and more base pair, the more and more lengthier the chromosome will be. So as you see clearly, the tallest is the chromosome 1. So chromosome 1 has highest number of base pair. Okay, historically, many years back, chromosomes were named according to the except the X and Y chromosomes, other chromosomes from 1 to 22 named according to their height mostly. Okay, except there's a little exception with now new technology. We know chromosome 22 and 21 is not goes according to the rule, because 22 is little bit higher, height is little bit higher than the 21. Except that small exception, other chromosomes from 1 to 22 is named according to their height. 1 is the tallest one, and then reduced. The height will be reduced from 1, 2, 3, 4, like inconsequently it has a less amount of height. In this picture, 21 and the Y little bit having a same amount of height. Okay, so X and Y is we have not considered when naming according to the height. Okay, there is something else we need to remember. The number of the base pair doesn't equal to the number of the gene. So some chromosomes which has comparatively reduced number of base pairs, that means comparatively its height is less, but they can be containing higher amount of gene, higher amount of protein coding regions. Okay, examples are chromosome number 19 and 11. 11 and 19 doesn't look like having a huge height compared to other ones, but they have high amount of gene. So the naming is according to the number of the base pair, not according to the number of the gene. So you don't call chromosome 1 have the highest number of the gene or 2 have the second highest number of gene. You don't call like that. It's mainly number of the base pair. Okay. Gene number of the gene is different. But in other hand, chromosome 1 have high number protein coding region. But it's for the chromosome 1. But for other chromosomes, we can't tally the chromosome number with the gene, because as I said, there are exceptions like chromosome 19 and chromosome 11 has high number of base pairs high number of protein coding genes compared to their surrounding other chromosomes. Like more than the 10, 11 has high genes, although 10 has high number of base pairs compared to 11. And when considering 19, 18 has higher number of base pairs more than the 19, but protein coding genes are higher in the 19. Okay. Other important things we can talk under this topic is about the trisomies. Okay,